CornerGenomics, genetic testing explained.
Genetic testing often reveals something called a variant of uncertain significance, or VUS. In this video, we'll put VUSs in context and show how they are approached differently in clinical situations like doctors' offices and hospitals versus in genomic research. A VUS is a genetic change where we don't yet have enough evidence to call it harmful, pathogenic, or harmless, benign. Our ability to sequence our genome is still fairly new, and genetic data at this level is noisy. The average person has millions of harmless genomic variants in their DNA. Genomic analysts use software to filter through and prioritize detected variants in order to focus on those that are most likely to be causing problems. ClinVar is a public database for genetic professionals to report variants and how they were classified. Here, a search for a particularly large gene reveals 36,000 reported variants. Looking closer, only 15% of these variants are known to cause disease, with the majority deemed likely benign or uncertain. Given the sheer volume of variation in a typical human genome, searching for a diagnosis is less like finding a needle in a haystack and more like finding one in a field of thousands. Thankfully, we have technologies, powerful filters, computational tools, and databases that act like magnets and help pull the most needle-like candidates to the top of the pile. Genomic tools and researchers apply broad evidence towards prioritizing these variants, or tuning the magnet, a subject for a future video. Another key factor is approaching genetic testing with a clear question. What are we trying to explain? What symptoms are present? The process of describing these symptoms is called phenotyping, and it is an important part of both clinical and research testing. Detailed phenotyping helps prioritize genes associated with the disease or traits in question. In a clinical situation like a doctor's office or hospital, testing must have a predictable timeline. Thus, only the variants that are most likely to be causing symptoms are listed on the report. Some of these variants may still be classified as a VUS, in which case clinicians typically don't base medical decisions on the variant. It gets documented, monitored, but not acted on. Regular visits with your clinician allows them to revisit variants, discuss interim health changes, and consider additional testing or reanalysis. Research teams aren't subject to the same time constraints as in clinical testing. They consider a larger batch of candidate variants and may do further investigative work if they think a VUS holds the answer or could lead to advances in genetic research. Researchers test variants in lab models, look for other individuals with the same variant, or publish case reports with collaborators around the world. They're able to grasp at straws, because given time and resources, these straws can lead to new discoveries and important diagnoses. So, what does this mean for patients and families? A VUS may not give immediate answers, but in research, it can be the seed of progress. By connecting with registries or participating in gene-specific research, clinicians and patients can help advance genomic knowledge and shorten the path to an answer for families in the future. Gene-specific research opportunities can be found on www.clinicaltrials.gov. Several rare and undiagnosed disease research studies are linked in the description below. In the next video, we'll break down variant nomenclature, what those C dots and P dots actually mean, and how to dive deeper on a particular variant of interest. If you'd like to keep learning how genetic testing works and how uncertain results become discoveries, subscribe here at CornerGenomics.